It's 10 seconds to 10, so I guess I can still say good morning, everybody. My name is David Sabernik. I am the head of business development and public affairs with the Medicon Valley Alliance. And on behalf of our Danish-Swedish Life Science Cluster Organization, it's a pleasure for me to welcome all of you, members and potential members, to this inspirational webinar titled Randomization and the Limits of Precision Oncology, organized with our member company IDDI. Today's speaker is, as I assume you already know, the medical director of IDDI, Everardo Saad, who will take over shortly. So I hope you have fastened your seatbelt and will enjoy the ride. If you get inspired not only by the content of today's webinar, which I know you will, but also by the concept, and start to wonder if it would make sense for your company or your organization to organize or co-organize an inspirational webinar with Medicon Valley Alliance, feel free to reach out to me. You will find my contact info on our webpage, and it will also appear on the final slide after today's presentations. I also encourage you to visit our webpage event calendar, where you will find a number of hopefully interesting and inspiring online and offline events organized either by Medicon Valley Alliance or by some of our member companies and partner organizations. I also encourage you to visit the webpage network section and see if there are any of our networks which could be of relevance to you. As an employee with an MVA member company, you can join any network and attend as many network meetings as you like, free of charge. And we actually had an online Medicon Valley Alliance oncology network meeting yesterday, which was recorded and is now available on Medicon Valley Alliance YouTube channel. Finally, a few practical remarks. It's pretty much standard operating procedure. But as you can see here on the slide, you are automatically muted. You should use the chat for technical questions only, and use the Q&A function for questions to our speakers. And as I just said, we normally record these events, and this is the case for this webinar as well. And you will receive a follow-up email with information and a link to the recording. I guess that's enough said on my part. I am not the reason why you tuned in today. So let's get started. And without further ado, over to our moderator today, Eric Falvey from IDDI. Eric, the screen is yours.
Thank you very much, David, and good morning to everyone, and thank you for joining us. My name is Eric Falvey, and it is my pleasure to be your moderator for today's webinar. The session is proudly hosted by IDDI. We're an expert center in biostatistical and integrated eClinical services for pharmaceutical and biotechnology companies in several disease areas, especially in oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels, Belgium, with the U.S. Center of Operations in Raleigh, North Carolina. So now it is my pleasure to introduce you to this morning's speaker, Dr. Everardo Saad. He's the medical oncologist and the medical director at IDDI, and he'll be sharing his thoughts on randomization and the limits of precision oncology. Everardo, the floor is yours.
Thanks, Eric.
Sure.
Thank you, David, and all the MVA team as well. And thanks everyone for joining us this morning in Europe. We have so many webinars these days, and I really appreciate you guys taking the time, and I hope you enjoy it. So I'm going to talk about randomization and the limits of precision oncology, and I hope to make it clear what I mean by that. So let me start off by saying that this topic can be, like many others, can be approached from different angles. at least three that I can think of from the point of view of drug development, clinical practice, and policy making. And I guess some of the confusion, or at least controversies that exist in this field, especially with regard to the merits of randomization in precision oncology, arise from the fact that there are different points of view. But I wanted to make it clear that I'm approaching this from the point of view of drug development, with a little bit more emphasis on early phases of drug development, phase 1, phase 1/2 trials, expansion cohorts, more than late phase trials. Although I'll give many examples of especially phase 3 trials that have been important in this field, but the focus is on early phases of drug development. But first, let's ask a very important question. What is a curmudgeon? So for those of us who are not native English speakers, I guess the webinar being at this time in Europe, I guess most people are not native speakers. It is a relevant word, and I'll say why. So Ian Tannock, who's one of the luminaries of medical oncology, wrote this very controversial article in the New England a few years ago, and he titled it Limits to Personalized Cancer Medicine, which is in a way very similar to the topic of this webinar. But in that paper, he was mostly criticizing the view that precision oncology is, let's say, ready for prime time, or ready to solve our problems. And in fact, he and his co-author present arguments for why this is not the case, especially because of the fact that at the time, it was estimated that only 8 to 10% of cancer patients would be eligible to truly personalized oncology or precision oncology, and also because of the price, because many of the drugs and interventions are still very pricey. So most of the critique was around that. And today, my focus would be on another aspect, which is more methodological, and it's related to clinical trials and its randomization. But in that paper, Tannock and Hickman said that the concept of personalized, or let's say precision medicine, is so appealing that seemingly only curmudgeons could criticize it. And then Google dictionary tells me that a curmudgeon is a bad-tempered person, especially an old one. So I'm saying that because I think you guys will not take me for a curmudgeon, because I'm criticizing, in a way, precision oncology, but I'm doing this from the point of view of methodology and the need for randomization. So that's the essence of the presentation. Now, what do I mean by precision oncology? I guess we could discuss the definition, but there's a very simple one and very, let's say, spelled out in even a lay person's terms. It is giving the right treatment to the right patient at the right time. So this, of course, entails in oncology using targeted therapy and using predictive biomarkers, like I will show in the next few slides. But I think as much as this is a valid definition, there are at least three implicit meanings. There is, of course, an overlap among these three meanings or angles, but I think they're sufficiently distinct that we can even provide examples, as I'll do in the next few slides. So there is the first connotation, which is choosing treatment based on predictive biomarkers. I think I could say this is the methodologist's mindset, let's say. In my view, it should also be the drug developer's mindset. But there are two other connotations. One is to interrogate tumor genomes to select among existing therapies. I would say that's more the clinician's mindset, as I will illustrate in a while. And then there is the policymaker mindset, which is to use molecular features to select evidence-based treatment. So let me illustrate what I mean by that. So I would say the first mindset of the using predictive biomarkers, or like I said, the methodologist's mindset is truly to base treatments on or treatment choices on randomized trials in which you have not only two competing interventions, hence the randomization, but also ideally you have biomarker positive and biomarker negative patients. And whether you have a qualitative interaction, which is the case here for gefitinib in mutation positive in non-small cell lung cancer, whereby it works quite well, better than chemotherapy in mutation positive patients, and even worse than chemotherapy in mutation negative patients, or whether you have a more quantitative interaction, which is the case for cetuximab or other anti-EGFR antibodies in colorectal cancer, depending on whether or not you have KRAS mutation. So this is the mindset that I'm denoting by more based on predictive biomarkers. But then you have the clinician's mindset, which is to think about patients individually, and then you see things like, considerations about how do you obtain tissue? Is it biopsy? Is it liquid biopsy? What site do you biopsy? What kind of platform do you use to interrogate the genomes? You have the need for bioinformatics. You even have the need for somewhat complicated algorithms to define, for example, how suitable patients are to different treatments depending on the level of expression of one or more genes and the availability of drugs. So in the end, you can classify patients, for example, like in the iPredict study, based on pathways that are deranged, and try to come up with drugs that match these pathways. And likewise, for example, in the Winter trial, which is not randomized, you can stratify patients based on their matching score. And so you can use this for individual, let's say, decisions, and try to see if this has indeed prognostic value. And the third mindset that I spoke about, which is more the policymaker, it's evidence-based choice of treatment, whether the evidence will come from, for example, a non-randomized phase 2 trial, which was the case for larotrectinib in NTRK positive tumors. So here you have a variety of tumor types that typically don't respond to other agents, and many of them have very deep responses. So this is now acceptable as level of evidence for drug approval, of course, in many cases pending confirmation, or whether you have evidence from conventional phase 3 trials in which a targeted agent is compared with chemotherapy, for example, after its initial accelerated approval. So again, this is all precision medicine, but I think there are slightly different points of view. Now, I don't think I need to explain to this audience what biomarkers are, but I just wanted to refresh our memories, let's say, about the definition of a predictive biomarker, which is used to identify patients who are more likely, if they have the biomarker, they're more likely than biomarker negative patients to have some kind of benefit from treatment. If you're talking about a predictive biomarker being used for treatment selection, arguably you can also use it for toxicity, but that's not what I have in mind now. So a predictive biomarker is truly something that can really distinguish between patients who will have benefit from those who will not have benefit, or, like I said, in the case of qualitative interaction, even might have harm from the intervention. And this is the ideal for precision oncology. Now, there is a list that the FDA publishes with the approved companion diagnostics, which is, let's say, the regulatory aspect of these biomarkers. Many here are not predictive biomarkers, they're just enrichment biomarkers, in the sense that they are useful for selecting treatment, but we don't know exactly if they are truly predictive in terms of demonstrating a treatment by biomarker interaction. But the fact is that currently, and this I did last week, there are 46 companion diagnostics approved or cleared, I guess, I should say. And 45 of those are in oncology. There's just one that's not in oncology. But it doesn't mean that there are 45 situations in which we have true biomarker drug pairs in oncology, because many of these companion diagnostics are dedicated to the same indication. For example, among the 46 or the 45 oncology cases, there are 11 which are for HER2, which by the way is an enrichment biomarker. So many, and likewise many others are repeated, for example, for EGFR in lung cancer, for PDL1, for example, for immunotherapy. So we don't have that large number of drug biomarker pairs. In fact, I counted on the list and could come up with only 16, let's say, pairs, not necessarily one biomarker to one drug, because in some cases, one biomarker to more than one drug. For example, for EGFR, you can use EGFR mutations, whether activating or resistant mutations, to select for many different tyrosine kinase inhibitors, and now even an antibody. But the truth is there aren't as many examples in oncology of really truly predictive biomarkers as there are drugs. So we have many more drugs approved and being tested than we have been able to validate biomarkers. And of course, immunotherapy is a notable example, perhaps alongside anti-angiogenics, of drugs that are out there for which we don't really have proof of predictivity. We know that some biomarkers can enrich for patients more likely to respond, but we don't know that they are truly predictive. And in fact, these authors did a systematic review a few years ago, and they found that the requirement of a predictive biomarker testing before prescription of an anti-cancer drug is often the result of development conducted only in biomarker positive patients. So in many cases, we cannot test for the presence of treatment by biomarker interaction. Now, precision oncology, of course, is very appealing. And many people have voiced, let's say, opinions about the fact that we should forego randomization, or we should it should be unethical to give to randomize patients. For example, if we have a highly active drug that on phase 1 trials are found is found to be to lead to high response rates, it may make future phase 3 trials lack equipoise. So it may make randomization truly difficult, especially, like they say, if you compare the new drug with true minimally active treatments that are in the market, or even placebo, of course. So this is, let's say, a worthwhile concern. And in fact, this has been done more or less at the same time as regulation was changing. And so the FDA has, for many years now, said that drug development is truly changing, in the sense that the previously existing clear demarcation between phases, let's say this is phase 1, phase 2, phase 3, with a different terminology. So this is, the demarcation is being blurred. So now there is a more seamless development of drugs. And also the other big change is the fact that efficacy has become a concern from initial development. And this has probably led or increased the number of cases in which phase 1 trials have expansion cohorts. So nowadays, many phase 1 trials of targeted agents are looking for early signs of activity, but in sufficiently large cohorts of patients to almost behave like phase 2 trials. And this has led to the publication of this guidance on expansion cohorts. And like many of you know, even to the approval of agents based on evidence that is somewhat different from used to be done in the past. For example, there is the famous approval of pembrolizumab for microsatellite instability high tumors. And this was based not really on one clinical trial, but a collection of trials, 5 different multi-center trials, all of them single arm, looking at different cancer types, some of which with very few patients represented, for example, 2 cases of prostate or breast cancer. And this has led to the approval of this immunotherapy for patients with the right biomarker. So this is what's happening in oncology nowadays. And to give a more recent example, this drug was approved a couple of weeks or months ago, sotorasib, and this is a very long unmet need in advanced non-small cell lung cancer. Many patients who were thought to have no actionable or druggable alterations had in fact a KRAS mutation, and for 20 years now, research on anti-KRAS drugs has been not very fruitful, but now we have this and other drugs coming up that are effective against KRAS mutant, a specific kind of mutation. And in fact, 37% of patients had a response, whether partial or complete. Median PFS was quite good, 7 months for this largely treated patient population. Most patients were in 2nd or 3rd line. And the median OS is certainly very, very good, very promising. It's about a year, which is not very common in this line of therapy for this particular kind of tumor. Not only were responses relatively frequent, but they were also quite durable in many cases. And the toxicity was there. About 70% of patients had any grade of treatment-related toxicity, which was 20%, let's say, grade 3, 4. So there is toxicity, but this was enough to lead to the approval of this agent, as far as I know, in an accelerated fashion, pending confirmation. Now, approvals based on non-randomized evidence are nothing new. So the FDA has forever said that you don't need to have a randomized trial to obtain approval in oncology, especially if you're talking about an accelerated approval. And so what's usually done is that the drug is approved, but pending confirmation. But this depends on the rarity of the disease, on the let's say magnitude of the unmet need, the magnitude of the benefit observed in comparison with historical controls. So there are many things that the FDA takes into account, safety, of course. And so in one of their published reviews, this one for drugs approved between 2005 and 2007, 37 of 53 indications were approved based on randomized studies, but the rest was not. So non-randomized evidence is clearly acceptable, and the sample size may be somehow somewhat small in some cases. For example, in one case, 18 patients, but typically the non-randomized studies are a bit larger. So non-randomized evidence is no major news, and in fact this happens in Europe as well. So these authors have done a systematic review some time ago. I don't remember the number of drugs. I really cut and paste only from A to C, but the list goes all the way to Z. Z in that case is for zinc, so it's not oncology. But most of the drugs on this very big list are for oncology. And so whether accelerated or conventional approval, both for FDA and for EMA, there are many situations where drugs are indeed approved based on non-randomized evidence. So this per se is not new. What I think is in a way somewhat new is the allure, let's say, of precision oncology, because what happens is that since you have a clearly identified rationale, and a drug that supposedly targets driver alteration in tumors, it's very tempting to um, to create a sort of circular reasoning, almost, in the sense that if you have a drug that is supposed to work, it has to work. But of course, it will be tested, but we cannot abandon randomization for many reasons, because basically observational evidence is always dangerous. So let me give you some examples of what I mean by that. So maybe some of you know that erythropoietin, that the red blood cell growth factor, was used for many years in oncology, probably more than a decade, until 2003, without much reservation, because indeed it was able to increase hemoglobin levels. And so the reasoning was that this was good for patients, not only in terms of quality of life, but for example, for patients undergoing radiotherapy, this would increase tumor oxygenation during radiotherapy, which is a prerequisite for the effect of radiotherapy. So it was used and approved until they really decided to put it to the test. So a randomized study was done comparing erythropoietin with placebo in head and neck cancer. And in fact, there was a statistically significant difference in overall survival, unfortunately favoring placebo. So that's, let's say, one of the big examples that one can use to illustrate the problem. I have to say there aren't many out there, but it's an old one, but it's just to remind us that randomization is still the definitive proof of the worth of any drug, no matter how much rationale you have. And you can do different types of analysis to illustrate that in an almost comical way, let's say. For example, these investigators from MD Anderson looked at patients who were taking their antiemetic drug, um, and who continued the drug or who discontinued the drug. And they found a statistically significant difference in overall survival for patients who continued the antiemetic. Of course, no one will argue this is the effect of the drug, but probably confounded by the fact that these patients who continued on the drug were sufficiently healthy to continue their chemotherapy, and that's why they probably lived better. But they did this as an illustration of the problems of analyzing registry data. For example, at the time they were criticizing some registry data on anti-angiogenics. So this is yet another way to illustrate. But more topically to our discussion here, we sometimes see this kind of evidence in which you have, for example, here patients with non-small cell lung cancer treated with targeted therapy versus those who had no molecular drivers. Most of them are KRAS positive, like I said before, and those who had a driver but could not receive targeted therapy. And you see, publications showing an improvement in overall survival based on observational, even retrospective data. But the authors here are very cautious, and they say that this should not replace any randomized evidence. It's just hypothesis generating. And of course, one may even discuss from other points of view whether this is the right way to go or not. But one of the problems in this kind of analysis, probably illustrated here, is what we call immortal time bias or guarantee time bias. So patients who are able to receive targeted therapy may not be comparable with those who are not. So we don't know actually what the real difference is between targeted therapy and no targeted therapy for these patients. So this is hypothesis generating. And in fact, to the best of my knowledge, the only published randomized trial of precision oncology is still the Shiva trial, published 6 years ago by the French group. And in this trial, they created an algorithm to define what drug would match a certain molecular alteration and randomize patients to receive that drug or conventional chemotherapy, having PFS as primary endpoint, progression-free survival, and there was no difference in terms of progression-free survival. So up to now, the only truly randomized trial of precision oncology is negative. It has been criticized for many reasons related to the quality of the algorithm that was created to define the drug. So this can be discussed by people in the field, and I think it's a valid discussion, but the point remains that as far as evidence, we still don't have that much evidence favoring a precision oncology approach versus a conventional approach. So why do we randomize? This is not new. This is in any textbook of medical statistics or even dozens of review papers mention this. So basically, we want to control for selection bias, and we want to control most of all not for known prognostic factors, which we can take into account to a certain extent in analysis, but mostly for unknown prognostic factors. And arguably in a very complex disease such as cancer, most prognostic factors at any time point, at any given point in time, are still unknown, so yet to be discovered. So the basic reason for randomizing is to control for unknown prognostic factors. That's the most important one, probably quantitatively. Now, of course, we also want to avoid the effects of stage migration. So as you go along, not randomizing patients, but selecting them using more sophisticated technology, you may end up selecting patients with a better prognosis. And so the effects that you attribute to your treatment improving over time may not be because of the treatment, may just simply be stage migration or even improved supportive care. In some cases, we have to have randomized evidence if we need to make firm conclusions about time to event endpoints. It's all very well to look at response rates in single arm trials, because if you have a very high response rate in a tumor that almost never responds to anything, this is truly noteworthy. But in terms of PFS or OS, you never know if it's the effect of the drug or if it's purely a selection of patients with a better prognosis. So unless you randomize, it's hard to make conclusions about time to event endpoints. There are other reasons. Unless you have a randomized trial with biomarker positive and biomarker negative patients, you cannot talk about prediction, true prediction. You can talk about the prognostic role of a biomarker, but you cannot talk about the predictive role of a biomarker without randomizing. If you really want to validate the predictive biomarkers, you have to have randomization. Likewise, you need to have a certain number of randomized trials if you truly wish to continue to validate tentative surrogate endpoints. So at least using the accepted methodology of individual patient data meta-analysis, you need to have a certain number of randomized trials of about the same question or similar questions. And finally, if you want to inform health economic assessments, ideally you must have randomized trials, because otherwise it's difficult to talk about any kind of comparison. For example, increasing the ICER, the cost effectiveness ratio, unless you have randomized evidence, it's difficult. Last but not least, let's not forget toxicity, because it's very tempting to think that a targeted drug is, as a general rule, less toxic than chemotherapy, for example. In a way, I would say it's mostly true, but not always. So for example, in the Shiva trial, the incidence of grade 3 and 4 adverse events was about the same, arguably nominally even higher with targeted therapy than with chemotherapy. But let's say they were at about the same. More of concern in a recent study in multiple myeloma, a randomized trial, the Bellini trial, venetoclax was added to a backbone treatment. So what you see placebo here is actually just the way to blind the study. So this was active treatment versus the same treatment plus venetoclax. There was an improvement in PFS, leaving aside the issue of non-proportional hazards here, but there was an improvement, statistically significant, but probably because there were more toxic deaths in the active treatment group, in the venetoclax group, there was a decrease in survival from the addition of the new drug. This did not cross the boundary for statistical significance because it's an interim analysis for overall survival, but it's sufficient reason for concern to remind us that without a randomized trial, we wouldn't know about this, and we would never know what the survival experience would be in the control arm, and so we might as well attribute any toxic deaths to the disease itself or to the seriousness of the disease. Without having a control arm, you're not even sure if you can attribute causality to the adverse events. Now, for all of these reasons, some years ago, we wrote this review paper just advocating for the need to continue to randomize in precision oncology. So again, trying to counter some of the voices out there in the literature, and remind people that randomization is still relevant. We discuss some different settings there, how this could be done, but anyway, it's just a review of the evidence at the time. Now, whenever you talk about randomization in precision oncology, especially if it's a more clinical audience, I dare say, there are always concerns and arguments against. I group them into three let's say baskets here. That's what I think in the end what they boil down to. Three different arguments. The first one is the ethical argument. So the argument goes, if we have a drug that is against a certain target and we don't have good treatments for the disease that expresses that target, is it ethical to randomize? I think it's a valid question, but this argument is always tricky because it can go both ways. You can argue that is it ethical, that perhaps it's unethical not to randomize, because as people have said in the past many times, we should always consider randomizing, and someone who was very prominent in the 70s and 80s, T.C. Chalmers, famously said that we should, in drug development, randomize from the first patient. So you don't know a new drug, you don't know how toxic it can be in human beings. So arguably, you don't need to do every single first-in-man, first-in-human study in a randomized fashion, but it's good to have in mind that perhaps we should always consider randomizing very, very early on. Now, the ethical argument can be discussed forever, but I think the idea, of course, is not to subject patients to unethical treatments. If you have a situation where there is clearly no standard, everything that's been done in the past is clearly ineffective, and you have a drug that in preclinical work or in very early phase 1 is marvelous, then probably you don't have to do a randomized study. But I would say that this is rare. This is relatively rare. Certainly much less frequent than people who argue against randomization would claim it is. So I don't think the ethical argument is sufficient to say, let's not randomize in precision oncology. Now, there's, of course, the biological argument, which, like I said before, it may be circular. So if you have a drug that's highly effective against the target and you have the target, why not randomize? But this is dangerous. We never know if the target is truly the target. We don't know about off-target effects. There are some rare cases in oncology when this was not the case. Iniprib is the most, let's say, well-known historically of a drug that was thought to inhibit a certain target and was ineffective in phase 3, eventually found not to be hitting the target that was thought to hit. So not necessarily we know enough about the biology of tumors, and so we should be aware of the tautology of the circular logic. Another way to approach this is the N of one, let's say, mindset. in a way, it's almost a fallacy. But if you take the N of 1 argument to the extreme, then I have to agree. If we come to a point where you know in real time for every patient what's happening to their tumor in terms of all the drivers and all the mechanisms of resistance, then it doesn't make sense to randomize. If you truly have precision medicine at a point where for every individual patient at a given time point, there is one drug, then you can't randomize, arguably, because no two patients will be alike. But we're far from there, and I don't think we're even going to get close to that level of understanding about tumor biology to be able to do that. So I think the N of 1 may be a valid thinking in clinical practice, but like I said from the beginning, my focus here is on drug development, and on drug development, I think we have to stay away from that argument as much as possible. And then there are the practical arguments about the number of patients you have to screen, but that's for any kind of study, for whether it's randomized or not, about the rarity of the indication. Again, the rarity of the indication has nothing to do with the statistics behind the sample size. It has to do with the feasibility of enrolling patients, of course, but nothing to do with the statistics. So you can, let's say you have 30 patients to enroll in a study. Arguably, it's better to do a randomized study, probably 2-to-1, 20 patients in experimental and 10 in control, than having a 30-patient single-arm study. So let's say as a general rule, this is better and probably in the long term safer and even less expensive, because obviously in one study it might be more expensive to randomize than not to randomize, depending on the statistical assumptions and whether you will formally compare or not groups. But in the long run, it's probably safer, although I don't think it can be proved. It's probably safe to say that in the long run, you're better off randomizing than not randomizing. Now, when I say randomizing, I don't mean placebo, right? So there is a long list of possible comparators. Standard of care is always valid if it exists. Treatment of physician's choice is a good alternative for when there is no standard of care, arguably even in early phase trials, although this might be better in late phase trials. A good thing is immediate versus delayed administration, for example, with crossover. And then different types of variations. So you can add the experimental to standard of care. You can add the experimental to treatment of physician's choice. You can have single agent versus combination. Arguably, you can even randomize to different dose levels if nothing else. And this is what was done, for example, in the large famous 1,200 phase 1 trial, 1,200 patients phase 1 trial for pembrolizumab. You can also randomize to different schedules or different durations, including randomized discontinuation, which was used a few years ago but didn't really catch on. Now, this is what's being done in the ongoing IMPACT2 trial. And so now we're going to finally have a second randomized trial of true precision oncology. So patients will be screened, and if they don't have, if they have a druggable alteration and no approved agent, they will be randomized to a highly targeted therapy, a targeted therapy versus a treatment that is based on molecular analysis, on matching scores and stuff like that. So this would be a true test of an individual drug for an individual patient, and then there will be crossover depending on toxicity or disease progression. So this is, in my mind, very close to ideal design that should be pursued more often. Now, in a recent review paper, we argued for that. So we argued for increased use of randomization, more specifically in early phases of development, because what we see in our work is that sponsors often are faced with this dilemma. Shall I finish my dose escalation and then run expansion cohorts in different indications, or shall I randomize? Especially when it's immunotherapy, because in immunotherapy, it's becoming very difficult not to randomize, as most of you will know. So you have the dilemma between possibly fewer patients and more efficient design versus a better estimate and more of the treatment effect and a more informative design. Of course, we will always argue for this one with due consideration to all the ethical and practical barriers. If they exist, of course, we cannot propose that. But whenever feasible, we like to propose randomized expansion cohorts. Now, why do we say that? Suppose you have in your design assumptions, you think the control arm will perform at a certain level of activity, and the experimental arm will be better. Now, pretend you only ran a single arm study, so you only have experimental arm, and you see something that looks promising because it's close to what you expected, or something that seems to be disappointing because it's lower than expected, or outstanding, but because it's higher than expected. So if you only have that, you will be disappointed or very, very encouraged, but without an internal benchmark, it becomes very difficult to know what's going on, because it might well happen that not only did your drug perform well, but the control arm also performed well. It might happen that your drug performed poorly, but the control arm performed even worse, and in other situations. So you can't really have a good feel for what's going on without an internal benchmark for comparison. Now, I'm not saying here that the comparison is based on formal statistical testing, because that would entail a true randomized phase 2 or even a phase 3 trial, depending on the assumptions. But I'm saying that you can informally compare. In most of the cases, we propose that patients are randomized unequally, for example, 2 to 1, 2 to experimental and 1 to control. So you have more, you collect more evidence on the new drug, and you collect less evidence on the old drug that you already know, but you still have the data, the results for the patients treated with the old drug, so you have a benchmark for comparison. Now, it's almost impossible to speak about precision medicine and randomization without mentioning the big elephant in the room, which is big data and its friend, real world data. So it's really hard to skip that. I wish I could, but I'll say very little about that. But it's you know, you always come back to the same discussion about randomization versus observation. And very prominent people recently wrote in this, again, controversial article in the New England Journal of Medicine, the magic of randomization versus the myth of real world evidence. Whether it's a myth or not, I don't know, but it's true that there is a big hype about these things. My suspicion is that part of this hype is due to the technology and the ability to have these data, but I'm not sure how they will help. No one knows. Arguably, SEER, the large population-based data from the US, are big. I don't know if they are big data in the sense of, you know, how much curation there is, but they are big data because it's about huge number of patients. And these authors here have published this comparison of evidence from randomized trials from the early breast cancer trials collaborative group in breast cancer about the worth of radiation therapy. And there's a meta-analysis of thousands of patients showing no difference in survival here. And whereas the SEER data would suggest otherwise, and in many cases, the SEER data suggests even the opposite of the meta-analysis based on randomized evidence. For example, as you increase the number of lymph nodes, There is less benefit from radiation here when you use mastectomy, for example, and here it's the opposite. So the trend is exactly in the opposite direction, and we could argue forever why this is the case, but again, it illustrates the danger of observational evidence when you have randomized evidence to replace it. Now, again, I'm not saying we should always randomize. There are reasons for not randomizing, and one of other luminary of medical oncology, Richard Schilsky, published this paper with his co-author outlining outlining some reasons for why in some cases, we do not need a phase 3 trial to approve a targeted agent. So, for example, if preclinical studies confirm that the target is really a driver, you can really assay that in a reliable way. The patients are selected based on the correct assay. You see high response rates when in situations where historical data are dismal. And so on and so forth, if this is a life-threatening disease or not. So no one can disagree with that. There are many situations where probably we can forgo randomization, but I would say they are the exceptions rather than the rule. So in conclusion, I would like to say that we will probably continue to have randomized trials. It's not difficult to prove that. Think about the next KRAS inhibitor. So now we have one that's approved, maybe another one will be approved, and eventually someone will come up with one that is supposed to be more effective. Then you will have to randomize against the old KRAS inhibitor. So don't we have dozens of studies of phase 3 studies of anti-HER2 agents? We do. We have now almost, I would say, dozens of studies of TKIs in non-small cell lung cancer against EGFR or ALK alterations. So we'll never get rid of randomized trials one way or another, especially if we have to continue to validate biomarkers and surrogates. Now, we will have to continue to rely on evidence from non-randomized data. Like I said, in some cases, it is wise not to randomize, but we have to always consider the magnitude of the unmet need, the strength of the biological rationale, how rare the disease is, and how reliable the new methodology to minimize bias may be. And by new methodology to minimize bias, I'm talking about some, for example, causal inference methods that are being proposed to address, let's say, the deficiencies of observational data. But I guess we have to answer the following questions in a near future. Is the seduction of improved technology like molecular biology and big data, is this sufficient to relinquish, to abandon time-tested methodology, for example, randomized trials? I don't think so. Can we safely replace randomized trials by other types of designs, such as synthetic controls, in silico trials, or causal inference methods? We don't know. I will not say that I don't think so, but we don't know. Can we reliably develop criteria to forego randomization? I don't know. For the time being, they are pretty much based on rationale, but are they predictive of the non-need to randomize? We don't know. Time will tell. I guess a very pragmatic question is, if we eventually have to randomize, is it better to randomize early or late? Because the further you go along in the development of a drug, the more you prove that it's truly truly active and seemingly more active than control, it becomes more difficult to randomize. So should we not randomize earlier rather than later? And I should say, as a medical oncologist, can I hope to reverse the title of this talk? So instead of talking about randomization and the limits of precision oncology, could I in the future come back and tell you about precision oncology and the limits of randomization? In other words, can we put limits to randomization rather than to precision oncology? I don't know, but time will tell, and I'm curious to know how this will evolve, and also curious to know what you guys think, and happy to take any questions. Thank you very much for your attention.
Hey, Everardo, thanks again for that presentation. I'm looking at the Q&A box here. There are a couple, maybe we can address these before the end of the session. So I'll begin. One is about biomarkers. The question is, do you propose always randomizing biomarker negative patients?
Well, always is a dangerous word. So ideally, yes, of course, for reasons that I outlined. I think ideally, yes, for in order to have true predictivity. In some cases, even regulatory agencies will want to have evidence on biomarker negative patients, at least to be able to tell that the biomarker is an enrichment biomarker. So I would say whenever possible, yes. So that's the short answer to that question.
Okay. Another one came in. Okay, what are the implications of this topic for immunotherapy?
Yeah, so immunotherapy is tricky in a way. I didn't mention immunotherapy many times, and I tried to stay in the realm of targeted therapy, because at least until now, it's more what I have in mind, at least when I think about precision oncology. In other words, really giving a drug based on a predictive biomarker. Obviously, many immunotherapy drugs, if not all, are targeted agents in the sense that they have a very specific target to which they bind. But in many cases, I would say in all cases, we don't really know if the marker is predictive. So if the target is truly predictive of the treatment benefit. In many cases, there have been analyses stratifying patients on the level in terms of the level of the biomarker, but it's not proven that we have true predictivity, for example, for PD-1, PD-L1 checkpoint inhibitors. But yeah, but in general, I would say, again, the short answer is yes. The same, I think the same reasoning applies to immunotherapy.
Okay, thank you, Everardo. Well, David, looking at the time, I think we're coming close on the hour, so maybe it's a good time maybe to kind of wrap up. I think that's it for the questions that we've come in so far. I guess if someone has additional questions, please feel free to send them to us, and we will certainly address them after today's presentation. But anyway, I think I can pass the baton back over to David to close out the session. Thanks again, Everardo.
Thank you, Eric. Thanks everyone for your attention.
Yes, and thank you very much, Eric. And then on behalf of Medicon Valley Alliance, I would like to say au revoir, auf Wiedersehen, see you later. I hope it's been time well spent and that you have appetite for more MVA events in the future. As mentioned earlier, feel free to reach out to me if you want to know more about this and other MVA event concepts. And if your company is not yet a member, and you want to explore the business case in a membership, I'm also the MVA go-to person. Finally, I would of course also like to thank Everardo and the entire IDDI team who made this webinar possible. And I guess with that said, we have reached our destination, and I will conclude this inspirational webinar. Have a nice day and take care out there. Bye.